Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Raval from Dr. APJ Abdul Kalam Government College, Silvassa, part of UT of Dadra and Nagar Haveli. Today, we are going to discuss about a disease associated with glycogen degradation, that is a part of glycogen storage diseases which we learn under the paper carbohydrate metabolism. So students, let's see what is there in this module for you. We will explore the concept of glycogen storage disease. We will study the glycogen storage disease associated with the glycogen degradation, and we will try to explore the molecular basis of the diseases. Glucose derived from the glycogen breakdown is an important source of energy for the body. It provides energy for the muscle contraction and serves as the primary source of energy for the brain. Healthy people maintain a relatively constant blood glucose concentration, that is about 5 millimolar, to support the tissue and the organs. We keep the glycogen in the muscles as well as in the liver. Glycogen present in the muscles is available only for the muscle contraction, while the glycogen present in the liver is required to maintain the blood glucose level of the body. Several metabolic enzymes are responsible for maintaining this balance, especially the enzymes that are involved in the glycogenolysis and glycogenesis. We get the glucose from the dietary sources also, as well as the gluconeogenesis that also helps in the maintaining the glucose balance. Under normal circumstances, they are tightly controlled by the organism. Allosteric control and covalent modification helps to mediate the enzyme activity. Glucose concentration is maintained with the help of hormones. Adrenaline, glucagon will also helps in maintaining the glucose concentration. Now, let's explore the glycogen storage diseases. Glycogen storage diseases are result from the genetic defect. This defect may be a mutation which causes this enzyme to become inactive. Which enzymes are being inactive over here? Those enzymes which are involved in the glycogen metabolism. Such defects are inherited and requires that both parents carry at least one defective gene. A zygote, whose parents are both heterozygous for a glycogen storage diseases, has a 25% chance of inheriting this disease. While each of these diseases prevents the correct production of a different enzymes, most glycogen storage diseases inhibits cells' ability to synthesize or metabolize glycogen, which creates a crisis of the energy. You think that if our body does not able to metabolize glucose or a glycogen, then what happens? Glycogen will not be available, glucose will not be available, and it creates a crisis of the energy. It has a direct effect on the liver and muscle tissues, which contain most of the body's glycogen deposits. As I told you that glycogen is present in the muscles as well as liver. Glycogen storage diseases affects primarily the liver, skeletal muscles, hearts, central nervous system and the kidneys. The principal storage depots of the glycogen are the liver and the skeletal muscles. With glycogen storage diseases, hypoglycemia is the primary indicator of liver involvement. Muscle cramps, exercise intolerance, muscle weakness, that is hypotonia, and fatigue are the typical of a glycogen accumulation in the skeletal muscles. These are the symptoms when the glycogen is getting accumulated. These things are observed in a patient. In addition, the peripheral and the central nervous systems, myocardium, and the renal tubules may also suffer from the aberrant glycogen accumulation. Early diagnosis and treatment are very important. Why early diagnosis and treatment is important? Certainly, we do not have a cure right now, but if we have early diagnosis and the treatment, then we may able to improve the life of the patient. Right now, we have a symptomatic reliefs. This early diagnosis and treatment are important for improving the quality of life, reducing the damaging effects on the organ, and that become engorged with glycogen and extending the patient's lifespan. More recently, enzyme replacement and gene therapy have been explored using animal models and skeletal muscle cells obtained from the affected parents. Glycogen storage diseases is a genetically inherited diseases which are characterized by deficiency in enzyme production along the pathway of a glycogen storage and breakdown in the liver. The different types of glycogen storage diseases are derived from exactly where the mutation occur on the glycogen gluconeogenesis pathway. Both it involves the both glycogen degradation as well as glycogenesis as well as gluconeogenesis. Do remember, we have four sources by which we can maintain the blood glucose, that is dietary, glycogen breakdown, glycogen synthesis and gluconeogenesis. In preparation for understanding this disease, we need to understand the basic biological concept such as how genetics play a role in the disease by producing a mutation that affect the glycogen gluconeogenesis pathway in the liver. The concept map of the class. Initially, we will try to study the glycogen breakdown. We will have a brief review on that. Then we will try to explore partially the importance of regulation, the biochemistry of different regulatory mechanism of a glycogenolysis. We will then study the disease associated with glycogen degradation. This is a structure of a glycogen. As you can see, glycogen is a highly branched large polymer of glucose molecule which is linked along its main line by alpha-1,4 glycosidic linkages. Branches arise by alpha-1,6 glycosidic bond at about every 10th residue. Glycogen storage diseases relates to a set of metabolic disorder occurs due to the defective glycogenesis or a glycogenolysis. Classification of glycogen storage diseases are according to the type of enzymatic deficiency. It is also classified on the basis of kind of primary organs involved. Glycogen storage diseases are usually diagnosed in infants or early childhood. Enzymes associated with glycogen storage diseases are involved in the regulation of glycogen metabolism. It is observed that there is phenotypic variation is observed when specific enzyme is altered by mutation. Such diseases concern primarily the liver, skeletal muscles, heart, and sometimes the central nervous system. These distinctive diseases are relatively diverse in age of onset of symptoms, morbidity, and mortality. A person with a glycogen storage diseases has a deficiency of one of the enzyme responsible for synthesis or a degradation of glycogen in the body. It results into abnormal concentration of glycogen in the tissue or imperfectly formed glycogen. In glycogen storage diseases, human body is not able to make enough glucose or not able to use glucose as a source of energy. The diagnosis of a glycogen storage diseases depends on an individual's symptoms. Glycogen storage diseases are genetic disorder. They are caused due to mutation in the genes related to the glycogen metabolism. It is detected in infancy or early childhood. In severe case, it is immediately identified, while milder types may not be identified or noticed for several years. General characteristic symptoms includes hypoglycemia, hepatomegaly, growth retardation, and irregular blood biochemistry. A more specific diagnosis can be done by analyzing concentration of glycogen in a biopsy sample or by assaying the sample of enzyme activity. Genetic diagnostic techniques are also available. Glycogen storage diseases are currently not curable. Generally, it is treated to manage the symptoms. Treatment involves organ transplant or dietary treatment. In future, it may be possible to treat such disease by using gene therapy. In this chapter, we will learn the glycogen storage diseases associated with glycogen synthesis. The diagnosis of glycogen storage diseases. It is detected in infancy or early childhood. It is immediately identified, while milder types may not be identified or unnoticed for several years. General characteristic symptoms include hypoglycemia, hepatomegaly, growth retardation, and irregular blood biochemistry. A more specific diagnosis can be done by analyzing concentration of glycogen in biopsy sample or by assaying the sample for enzyme activity. Genetic diagnosis techniques are also available. Glycogen storage diseases are currently not curable. Generally, it is treated to manage symptoms. Treatment involves organ transplants or dietary treatment. In future, it may be possible to treat such disease by using gene therapy. In this chapter, we will learn glycogen storage diseases associated with glycogen synthesis. Now let's have an overview of glycogenolysis. Glycogen phosphorylase catalyzes phosphorylated cleavage. It occurs at the non-reducing terminal of the glycogen chain. Debranching enzyme separates the branch and transfer it to the main chain. It also releases the residue of at alpha-1,6 branch as free glucose. Phosphoglucomutase converts glucose 1-phosphate into glucose 6-phosphate. Glucose 6-phosphate can enter into glycolysis. In endoplasmic reticulum of liver, glucose 6-phosphate can be changed into free glucose by glucose 6-phosphatase. Free glucose is released into blood. Now, let's try to explore the disease associated with the glycogen synthesis. Actually, the disease associated with the glycogen synthesis and degradation are classified under the group called glycogen storage diseases. So over here, we will review the diseases that is associated with the glycogen synthesis. That is a glycogen storage disease type 1, glycogen storage disease type 2, glycogen storage disease type 3, glycogen storage disease type 5, glycogen storage disease type 6 and glycogen storage disease type 10. So in short, 1, 2, 3, 5, 6, 10. These are the diseases that is associated with the glycogen synthesis. First, we will try to understand the glycogen storage disease type 1. It is also known as von Gert's disease, hepatorenal glycogenosis, type 1 glycogenosis, glucose 6-phosphate deficiency glycogen storage disease. Their general symptoms includes enlarged liver, that is hepatomegaly, and hypoglycemia, that is low blood sugar. It is observed as an autosomal recessive disease. It is mainly of 3 subtypes: glycogen storage disease type 1A. Over here, there is a deficiency of glucose 6-phosphatase. Glycogen storage type 1B, there is a deficiency of glucose 6-phosphatase transporter. While glycogen storage type 1C, over here, there is a deficiency of transporter. Chromosomal location of the respective gene are given in this slides. Glycogen storage disease diagnosis includes the genetic studies, measurement of a size of a liver and kidney, hematology studies like blood glucose, cholesterol, triglycerides, lactate and uric acid. Treatments includes glucose solution is given intravenously or regular oral raw cornstarch at the interval of 3 to 6 hours. Liver transplantation or a renal transplantation. Promising results are observed in animal model using gene therapy. In future, it may be useful for human treatment. Glycogen storage disease type 2. It is also known as POMS disease type 2 glycogenosis. It occurs due to deficiency of lysosomal 1,4-glucosidase, which is also known as acid maltase. Lysosome carry out digestion of cellular components along with the digestion of glycogen, maltose and other oligosaccharides. Due to deficiency of acid maltase, there will be increase of undigested glycogen in lysosome. It affects the functioning of lysosome. Lysosomes are found in almost all types of tissues. This will affect the functioning of all major organs, particularly the cardiac tissues. POMS disease is a deficiency in the lysosomal 1,4-glucosidase, which is also known as acid maltase, which causes a buildup of undigested glycogen in the lysosome. You just remember the friends, glycogen breakdown occurs at 2 places, one by the process called glycogenolysis and another in the lysosome. Lysosome contains the acid maltase which helps in the breakdown of glycogen. If there is a deficiency in the acid maltase, then it results into the POMS disease. Lysosomes are normally responsible for breaking down cellular components such as proteins and nucleic acids, but they also take up and undigest glycogen, maltose and other oligosaccharides. The interior of a lysosome is typically more acidic than the cytosol. Lysosomal enzymes are more active at acid pH than at neutral pH. Hence, the name acid maltase has been given to this particular enzyme. Glycogen builds up, prevents the lysosome and the rest of the cell from functioning properly. Since this particular enzyme is absent, obviously the lysosome is not able to digest the glycogen. So ultimately there will be an accumulation of the glycogen within the lysosome and which impair the function of the lysosome. Since lysosomes are distributed throughout all types of the tissue, all major organs and especially the heart are getting affected. There are two forms of the type 2 glycogenosis, and they are distinguished by the age of onset. The infantile form and the disease is severe. Affected children generally do not survive past 24 months, with heart failure being the usual cause of death. Therapy for this patient can only be palliative. The adult form is not always fatal, with respiratory failure being the biggest cause of the death. The diagnosis of the infantile form can be made at the birth. The heart is usually large. Cyanosis episodes of dyspnea occurs early. Glycogen storage disease type 3. It is also known as deep brancher deficiency, Cori disease, Forbes disease, limit dextrinosis. It occurs due to deficiency of glycogen debranching enzyme. Glycogen debranching enzyme helps in the removal of branches in the chain of glycogen. Deficiency of glycogen debranching enzyme results into the partial breakdown of glycogen. It produces glycogen with short outer chain. Such partially digested glycogen accumulated in organ tissues. It causes damage to the tissue of liver and muscles. Their general symptoms is hepatomegaly and hypoglycemia, hypotonia, small stature. It is observed as an autosomal recessive disease. Debranching enzyme is synthesized by the gene amylo 1,6 glucosidase 4 alpha glucotransferase gene. It also has two subtypes, that is 3A and 3B. Glycogen storage disease type 5. It is also known as McArdle disease, type 5 glycogenosis, myophosphorylase deficiency. It occurs due to deficiency in muscle glycogen phosphorylase. It is also known as myopathic glycogen storage disease since it affects mainly muscle tissues. Glycogen phosphorylase is also found in the hepatic tissue. Glycogen is broken down into glucose 1-phosphate glycogen phosphorylase. It is carried out by phospholysis reaction. Glycogen phosphorylase will act repeatedly on the non-reducing end of a glycogen chain. Glycogen phosphorylase can act continuously until it reaches 4-glucose away from the alpha 1,6 branch point. So, it has a certain general symptoms, that is muscle pain, muscle cramps, tenderness in muscles and exercise intolerance. It has a different forms. It is rapidly fatal in neonatal form. It is in mild form with congenital myopathy. Glycogen storage disease type 6. It is also known as Hirsch disease type 6 glycogenosis. It occurs due to deficiency in liver glycogen phosphorylase. Glycogen is broken down into glucose 1-phosphate by glycogen phosphorylase. Its general symptoms are again the hepatomegaly and hypoglycemia. Diagnosis includes the genetic studies, the quantitative analysis of glycogen phosphorylase in liver cells, microscopic studies of liver tissues. Treatment includes the dietary treatment, gene therapy may be possible in future. The glycogen storage disease type 10. It is observed due to the deficiency of cyclic 3,5-AMP dependent kinase. It is an autosomal recessive disorder. This disease directly does not relate to the glycogen degradation. In fact, it affects the regulation of glycogenolysis. Gene of cyclic AMP dependent kinase is located on the chromosome 17q23-24. That is all visible on the screen. During regulation of glycogenolysis, cyclic AMP act as a second messengers. Their symptoms includes the asymptomatic hepatomegaly. Glucagon or adrenaline does not stimulate the glycogenolysis in such cases. The diagnostic part having the microscopic studies of the liver biopsy, enzyme analysis and DNA-based genetic testings. So ultimately, a glycogen storage disease affects primarily the liver, skeletal muscles, hearts, sometimes the central nervous system and the kidneys. These unique diseases are quite varied in age of onset of symptoms, morbidity and mortality. Glycogen storage diseases are classified according. So students, let's try to conclude what we have learned in this module. In this module, we discuss the diseases associated with the glycogen. We try to explore the diseases of glycogen storage, specifically which is associated with the glycogen breakdown. We learn about the diseases associated with the glycogen breakdown. These are complex, rare and varied diseases that until the last decade were managed symptomatically. Current treatment is still directed towards lessening the symptoms, but there is an effort to prevent the effect. Many defects in glycogen metabolism have been identified. These are all related to a deficiency in enzyme activity at one point or another in the metabolism of glycogen. So students, the introduction of a recombinant enzyme replacement for infants with glycogen storage disease type 2 has shown a promising results. So in future, if we use the enzyme replacement therapy or a gene therapy, will come out with a solution. Several systematic studies have demonstrated the effect of enzyme replacement therapy on glycogen storage diseases type 2. Thank you, my friends.